Hey guys, welcome back to Bioinformatics with R. Today we're going to be doing a little bit of phylogeny type work. So we're going to be doing variant calling, and this is a valuable bioinformatic task where we take alignment of high throughput sequencing through BAM files, and we can compute a list of variant positions. So these are essentially SNPs or indels in the genetic code. And there are a lot of tools that can do this. There's online tools. However, Bioconductor, the biology suite for R, is able to do this from beginning to end in a very fast, efficient manner. So we'll be using that to take BAM files and identify variant SNPs or indels within a genetic code. So let's get to work. Okay. All right, so first thing we're going to do, let's load the required libraries. So there's a few of them that we need. So we want genomic ranges. We want, let's spell this right, GMAP-R, R-track layer, variant annotation, and variant tools. Let me clear all this stuff out here. And I'm going to make this a little smaller because it's kind of obnoxiously big. Okay, so let's load these. And while those are loading, we can say now we want to load our data sets. We need .bam and .fa, which are FASTA files, files for this pipeline to work. So we're going to say our BAM file, make a variable, and we're going to say file path get WD, and then we're going to do hg17/snips.bam. So this, even if you're doing Praxis, this is the code you want to do. So we have our, hopefully you've copied your hg17 SNPs. So this is a BAM file of chromosome 17 from humans. And we need the working directory for where it is. So if we run this, you'll see that we have, like, I'm doing this all in Praxis right now. So home, student, desktop, classroom, my files, and then the name of the BAM file. So we need to do the FASTA file too. So this would be essentially the same thing. Get WD, and the name of our FASTA file is chromosomes CR17 chromosome 17.83k.fa. Make sure it's in parentheses like that. Okay, so we have our two file paths. So annotation. Now we need to set up the genome object and the parameters object. Okay, so now what we need to do is kind of set up our objects, or get them into a proper form, I guess is the best way to put it, for this workflow to work. So we're going to say FA is R-track layer, and we're going to say FASTA file, and then we're going to give it our file path, FASTA file. Let's see if that works. Okay. So now you can see if we click on this, we have our FASTA file loaded in. Now we're, let's annotate again. Now we create a GMAP, genome object, which describes the genome to the later variant calling function. So this is just going to set it up as our data as a GMAP file, GMAP genome file or object, which has to be in that format for the variant calling tools that we're going to use later to actually be able to read it, right? Because these packages, they need to know where the data is. They're not intuitive in that they can't search for it themselves. They have a certain place they're going to look. So we have to have the data in that format. So we're going to say genome is our GMAP R, GMAP genome FA, and create equals true. So we need this function to create this object for us. So that's why we put that in there. And it's going to read the FA, the FASTA RTREC layer file that we just created. Okay, so it is still working. This might take a minute or so to run. There's a lot of bases in a chromosome. So imagine if you're doing an entire genome, this can take even longer, right? So we're only doing one chromosome for purpose of demonstration because we'd be sitting here for a while. If you were to be doing this and you wanted to do a whole organism, you could either sit and wait, or you'd have to use some sort of cloud virtual machine, kind of like this is, this Praxis environment, but you'd have to allocate more computing power to it. So as you can see, it's done. So now, let me see where we are. So this next step sets our parameter for what is considered a variant. So you have to define to these functions what you consider a variant, right? And so I'll start a new line for this next part. There can be a lot of fine tuning to this function. And so there's not a hard and fast set of rules or default features or settings, I guess are the best word, that will get you your proper number of variants for every organism, for every sequence, etc. So this is something that if you do this in the future, you might have to look into the literature a little bit to see what other people have done, or you can play around to see how to increase your number of variants, but you don't want to do it to the point where you're falsely identifying. So it's better to be more constrictive and identify less variants than it is to falsely identify more variants, if that makes sense. Is there anything else I wanted to add here? So we, for purpose of demonstration, are just going to use standard settings. So we will identify some, possibly not as many. And one thing that you could do too is if you were doing this for a research purpose, you could get your BAM files and purposefully have a test set of BAM files where you make variants yourself. So you just go in and manually change some bases to make SNPs, and then see how good this is at identifying them, because you'll know ahead of time, right? So it's kind of like a training and a test data set where you set up this script, you have a BAM file that you know where all the indels are because you purposely put them in there, and then see how good this is at identifying them. And you could theoretically then fine-tune your function here to identify what you consider as acceptable, right? Okay, that being said, we are now, where are we here? So we're going to say quality params, so qual params is tally variants param, and then genome equals genome, and then minimum map q, so our quality score equals 20. Can I run this up here? Genome? I did. Okay. So I'm gonna run this. I don't need that there. And then variable params, or variant param, sorry, is variant calling filters. Read count equals 19, so there's got to be 19. Yeah, let's do this real quick. So is this not the right one? Description, call variants. No, we want variant calling filters. Oh, let's try this again. Oh, it's not gonna let me pull up the thing. So this for calling variants. So our p value is how significant our variants are as identified by the statistics there. Oh, here we go. So at least this many high quality reads with the altered base. So it's going to look through all of the reads that we have, and it says it's only a variant if we have 19 of those reads present in our BAM file. And so the reason you would set this is because there could be sequencing errors where like a couple of sequences just miscalled the base pair. So we want enough of the sequences, so we dictated 19. You could change this if you have, higher depth of coverage for your sequencing or lower depth, you can lower it. But we're saying at least 19 of them have to have this variant for it to be considered a variant. And so the p lower is the lower bound of the binomial probability for a true variant. So this is just essentially setting our p value. Okay. So now we use call lowercase variants function in accordance with our parameters we defined above. So these are just setting the parameters, right? So we have our genome and our read quality, and then our read total count and the p-value. Now we actually have to do the variant calling. So we're going to say called variants is call variants, and we're going to read in our BAM file, and then we're going to use our quality parameters that we defined above, and our calling filter is going to be our variant params that we had above. So now you can see why we called those. So this might take a minute, so it's going to go through that BAM file now, and it's going to use our quality parameters and our variant parameters, and it's going to look and try to identify variants, SNPs, indels within this BAM file of our reads from our head throughput sequencing data. So I might have to edit the video here and fast forward because it might take a minute. But so some of the things that you can do with this, you can use variant identification of SNPs for things like phylogenetics, which we'll do shortly, because through evolutionarily, more closely related organisms should have more SNPs in common, right? Just their whole genome should have more in common, but the SNPs should be inherited because there isn't enough evolutionary distance for those SNPs to have been mutated to something different. And the other thing that you can do, error. Did I not call this first? Did I call qual params? I did. Oh, so I just didn't run var params. The other thing you can do, if you're more into kind of human genetics, is you can use SNPs for genome-wide association studies, which we're going to do in a later section here. And so the theory behind that is you can identify SNPs that are tightly linked, so they're very close in location to a gene of interest. So what you have to do is you end up getting a SNP map for your entire genome using these, a variant calling type pathway or workflow, I guess is the best word. And then what you can do is you pick a quality or a quantitative trait. So you could do height of people, or you could do, length of your finger, anything quantitative. And then what you do is you take a whole bunch of people and you have all of their SNPs. And theoretically, if a SNP is more present in your population that have long fingers, for example, then that segment of the genome where that SNP is present would likely be associated with the gene of interest. And so we'll get more into it when we get into the GWAS stuff. I just wanted to fill the time of that function running. But SNPs are important, so they're very useful tools. Okay, so now we're going to look at the head of our called variants. Okay, and so I'm going to drag this up so you can see a little bit. So it says we have identified 6 variants, 6 SNPs or indels within here. And as you see, it also gives us our reference and what our alternate is. So at this position, in segment NC, this is chromosome 17, you see we had like 759 reads. Our minimum was 19. Our reference, the most common SNP was a G, but the alternate was a T. We didn't have sample names in there. So our read depth, this is how many of our variants we had. So we had 20 of the T's. We had 22 of the T's at this position. So this is the total read depth. This is the G count, and this is the T count. So that should add up to that. So you get the idea. It gives you a lot of information of where the SNPs are, and the number of times it was observed, etc. Okay, so let's keep going though, because we can do more interesting stuff than that. Let's say we have identified 6 variants. Now we move on to annotation and load the feature position information from GFF. So now we want to see if those SNPs are in genes, right? And evolutionarily, you're going to identify many more SNPs that are not in genes, right? So in intronic regions or in intergenic regions, so the space between genes, because there's no selective pressure to prevent that. So if you have a mutation in a gene, it's likely, or possible at least, that you break the gene and then that organism dies because it was an essential gene, right? If you mutate in an intron or in an intergenic region between genes that doesn't code for a protein, there's not a lot. There can be selective pressure because you can have transcription factor binding sites and things like that. But for the most part, there's not as much pressure where that organism might die from that mutation. So we want to map these SNPs to see if they're in genes, right? Because those are, what we're more interested in. So we're going to say get annotated regions from GFF. We're going to make a function where we give it a file name. And then our function, we're going to say is GFF rtrack layer import GFF of our file name, and then convert it to a granges object. So all this function is doing is saying, okay, give us a file name. That file name, we will use the import GFF function to save it as GFF. So we're going to give it a GFF file name. It's going to use this rtrack layer import GFF to import it, and then we're going to convert it from a GFF to a granges file because that's what our variant calling stuff needs. So it's nothing too fancy, just an easy way to, whoa, this got big, to import our data as a GFF and then convert it to a granges. So note this info. Oh yeah. So you don't need GFF files. You can also, a common output file from sequencing pipelines is a bed file. You can also load this data from a bed file. So if you are using this in the future, I think it's rtrack layer import bed instead of import GFF. That's the only difference. But they both have the same data in it. Okay, so now let's get our list of genes from our GFF file. And we're going to use the function get annotated regions from GFF, and then we're going to say chr17.83k.gff3. Okay, so we have our granges object, which shows us where the genes are. Now we can calculate which variants overlap which genes on our chromosome. So we're going to do overlaps, genomic ranges, find overlaps, and then we're going to give it the GFF file. 83k.gff3. Okay. What did I spell wrong? Error in function, blah, 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 blah. Oh, did I not run? I probably had to run. Did I run genes? I did. Able to find an inherent method for function. Let's take a look here. The sequences all appear to be there. Oh, well, we don't want this. We need called variants genes. So I was trying to say find an overlap between a GFF file and nothing. So what we need is overlaps, genomic ranges, find overlaps between the variants that we found, which we identified up here, are called variants, and our genes that are from the GFF file. There we go. Okay. And now if we can print overlaps, and you can see that we have our overlaps identified. Let's see. query hits and subject hits. So this is just showing the number of hits between our variants and our genes. So let's do, and lastly, we subset the genes with the list of overlaps. Okay, so now if we do genes, subject hits, overlaps. Okay, so now we can see we have a lot of information here. Let's save this as a vector array. Let's do this. Identified. Okay, so these are, maybe it's not easier to look at that way. Let's pull this up. It gives you a lot of information, right? So we have our sequence names, the ranges. So this is the base pair ranges. Source, doesn't matter. This is what was identified in the GFF as having a mutation. So we have a non-coding RNA gene. So this is an RNA that's created but doesn't create a protein. We have a long non-coding RNA. We have an exon, so this is in a protein. A non-coding RNA gene, long non-coding RNA. Okay, so this is just printing out the same thing below. We have our transcript ID, so this is like information that you can use in Ensemble or something of that nature. The gene name. This kind of prints out kind of funky, huh? Maybe I probably could. Nope, still not going to do anything by doing that. If it's a novel transcript or not. So if we had stuff that, so this is a novel transcript, right? So this is something that has not been characterized before in our GFF file. The other ones are not novel transcripts, so it's something that is known. What else do we got here? The exon ID. So for the one that is an exon, exons have their own IDs as well, because you get things like alternative splicing, where different proteins use the same gene to put together proteins, but they use different combinations of those exons within that gene. So yeah, so that's it. So that's how we identify variants. Theoretically, where did my console's all messed up now. So theoretically, what you could do is save all this to like a VCF file, and then use it for phylogenies and things of that nature. So I'm rambling on a little bit here. Admittedly, I'm familiar with variant calling, but I'm not by any means an expert. So there's a lot more that you could dig into this. I just want you to have the exposure to it and kind of see what is possible. But we will use these identified variants like we just did in creating trees and things of that nature going forward. So I hope you liked the video. If you're interested in more variant calling phylogeny stuff, let me know, and I can look into learning some more pathways and things like that nature. But like I said, I hope you enjoyed it. We'll start working with phylogenetic trees, which I'm a little more familiar with, and we will go from there. So I hope you like this video. I'll see you in the next one.